Niacin, long recognized for its cardiovascular benefits and lipid-lowering effects, is increasingly being understood as a key regulator of intracellular energy metabolism, particularly through its influence on NAD, biosynthesis, and calcium signaling. Beyond its systemic actions, niacin enters a deeper metabolic conversation within the cell, modulating energy status, mitochondrial resilience, and lysosomal function. This positions niacin not only as a therapeutic agent for atherosclerosis, but also as a broader metabolic modulator with potential relevance in aging, neurodegeneration, and inflammatory disease. At the center of niacin's intracellular activity is its role in supporting nicotinamide adenine dinucleotide (NAD) metabolism. Niacin serves as a precursor to NAD, a critical coenzyme involved in redox reactions, DNA repair, and signaling pathways governing cell survival, senescence, and energy production. While several precursors, such as nicotinamide riboside (NR) and nicotinamide mononucleotide (NMN), are being explored for their ability to boost NAD. In aging and disease contexts, niacin's route is unique. It acts through the salvage and Price handler pathways and engages CD38, a multifunctional enzyme with dual roles in NAD, metabolism, and calcium signaling. CD38 catalyzes the conversion of NAD into NADP, nicotinic acid adenine dinucleotide phosphate, a potent second messenger that triggers the release of calcium from lysosomal stores through TRPML1 channels. This calcium mobilization not only affects lipid trafficking and cholesterol efflux, as seen in macrophages, but also has broader implications for mitochondrial function. Lysosomal calcium release has been shown to regulate mitochondrial bioenergetics by coordinating mitochondrial fission, fusion, and oxidative capacity, especially under stress conditions. Thus, niacin, by boosting CD38 expression and NAD levels, indirectly supports mitochondrial adaptation to metabolic demand. Increased lysosomal calcium flux enhances communication between organelles, including the endoplasmic reticulum and mitochondria, facilitating calcium transfer that sustains ADP production and prevents mitochondrial collapse during oxidative stress. This interorganellar crosstalk is vital in cells with high energy demands or those exposed to inflammatory stress, such as immune cells, neurons, and cardiomyocytes. Furthermore, niacin's influence on NAD levels may directly activate sirtuins, NAD, dependent enzymes that regulate mitochondrial biogenesis, autophagy, and stress resistance. SIRT1 and SIRT3, for example, enhance mitochondrial efficiency and antioxidant defense while promoting fatty acid oxidation and glucose utilization. By supplying the NAD needed for sirtuin activity, niacin helps maintain mitochondrial integrity, reduce RLS production, and enhance metabolic flexibility, all of which contribute to long-term cellular resilience. Importantly, niacin appears to function not just as a passive precursor, but as an active orchestrator of intracellular metabolic balance. It couples NAD metabolism with calcium-dependent signaling, providing a dual signal for energy optimization and stress response. This is particularly relevant in pathological conditions where mitochondrial dysfunction is central, such as in neurodegenerative diseases, metabolic syndrome, and chronic inflammation. Moreover, niacin's ability to reduce lysosomal cholesterol accumulation has downstream benefits for autophagy, a process tightly linked to cellular energy status. Lysosomal integrity is essential for autophagic flux, and by preserving calcium dynamics and lipid balance within the lysosome, niacin supports the degradation and recycling of damaged cellular components, further fueling mitochondrial performance and cellular repair systems. In sum, niacin is far more than a vitamin or a lipid drug. It is a dynamic metabolic modulator that intersects NAD, biology, calcium signaling, and mitochondrial health. Through CD38 activation and NAP-driven lysosomal calcium release, niacin promotes efficient cholesterol trafficking, enhances cellular energy capacity, and supports the stress-adaptive machinery of the cell. As such, it represents a promising candidate for broader therapeutic applications, not only in atherosclerosis, but across a spectrum of diseases rooted in metabolic and mitochondrial dysfunction.